Dr. Hume. Dr. Robert Hume is going to be talking about the Baird Institute's research into novel degradable blood vessel replacements. And with the money raised at our last appeal, we'll now be able to interrogate each individual cell that has grown into the artery replacements using this cutting-edge technology we referred to called spatial transcriptomics. And we'll be able to investigate how closely their gene expression mimics that of a healthy artery. But Dr. Hume will be presenting on this fascinating research today. I'd like to welcome both Professor Paul Bannon, who's chair of the Baird Institute, and also Dr. Robert Hume, lead of translational research at the Baird Institute. Paul will be just giving a brief introduction to this vital research, and then Rob will present on their work and their current findings. Both will be available for a Q&A at the end of the presentation, so keep that in mind. Just to introduce both Paul and Rob, Paul's a cardiothoracic surgeon of international standing with clinical appointments at Royal Prince Alfred, Concord and Strathfield Private Hospital. At Royal Prince Alfred, Professor Bannon is the co-chair of the Institute for Academic Surgery and director of the Robotic Training Institute. At the University of Sydney, he holds the professorial chair of cardiothoracic surgery and the Bosch Chair of Surgery. He is also the current head of the discipline of surgery for the Sydney Medical School and the academic director of the Translational Research Facility or Hybrid Theatre at the Charles Perkins Centre. So he doesn't have much on. Dr. Hume is a research fellow at the University of Sydney and lead of translational research at the Baird Institute. He undertook his bachelor's and master's in biomaterial science and tissue engineering at the University of Sheffield in the UK and his PhD at the University of Cambridge in the UK, before moving to Australia to complete his postdoctoral training. His work has spawned novel intellectual property and a myocardial infarction therapy currently tested for translation to the clinic. Dr. Hume has led major translational projects and lead author publications in high-impact journals. So just before I hand over to Paul and Rob, just a little bit of housekeeping. As I mentioned, all microphones have been muted just to make sure the webinar goes smoothly. Cameras have been left on, but for those who've just come in, if you feel free to turn them off if you would prefer. If you've got any questions throughout the presentation, just note them down to ask at the end, or just use the Q&A button at the top of the webinar page to send a question through during the presentation, and we'll get to them at the end. So, Paul, over to you.
Thanks, Catherine, and welcome everybody. I think this is exciting, having a webinar, one of my favourite topics, building bits of the heart. And the topic here, novel degradable blood vessel replacements, we've been working on for some time, but this group has really come together very nicely more recently with Rob joining the team, and it's allowed us to put together the Centre for Heart Failure and Disease of the Aorta, which is really a culmination of our efforts to join our clinical work, as in our hospital work, or our, what you know of as our surgery, and the translational side, where we base a lot of that work in the Charles Perkins Centre in the University of Sydney. Rob heads the translational group for the Centre for Heart Failure Disease of the Aorta, supported by the Baird Institute. So it's really his baby, but it works. It's based on a lot of the work and the interest of the clinical side that we have over many, many years, replacing the aorta or indeed building a better blood vessel to help in the long term. So Rob, I'm looking forward to hearing it as well.
Awesome. Thank you so much for that introduction. And thank you, Catherine, for that introduction. That's really appreciated. I guess it kind of, I'm going to go through this first slide, but essentially they've already kind of said everything for me, which is very polite of them. Yeah, I started my time at the University of Sheffield in the UK, went on to Cambridge, did my PhD there. Then I went on to the Westmead Institute over at Westmead and Westmead Hospital with James Chong, who's a clinician scientist and a cardiologist. And then, as Paul just said, I joined the Centre for Heart Failure and Disease of the Aorta within the Baird Institute. I'm working within the University of Sydney as well. And it's there that I work very closely with Paul, Associate Professor Sean Lal, Professor John O'Sullivan, and Cass as well, who I work in the lab with a lot. So what do I research? What am I looking at? Well, there's two main areas of research. One of them is this vascular regeneration that Paul introduced then, where we're trying to regrow damaged arteries and regenerate damaged arteries. And the second is cardiac regeneration and trying to regrow heart muscle that's been damaged following a heart attack, otherwise known as myocardial infarction. And so you can kind of see that over here. This is a microscopy image where we've stained the different proteins, different colors. And we've got a heart muscle cell here in green with the striations going across it. And the nucleus of that actual cell here in blue, which contains the DNA, which enables the cell to make proteins. And this red marker here, which is showing that that cell is actually going through division and trying to replicate itself, which is a very novel, new, exciting finding. But enough of that. That's not what we're here to talk about today. We're going to be talking about the vascular regeneration and growing damaged arteries. And maybe another day we'll talk about cardiac regeneration. Okay, so what causes damage to an artery? Like, how do arteries become damaged? And there's a whole range of different ways that can happen. One that is quite common is that you have plaque buildup within your arteries. And that can be in one of the coronary vessels, like within the heart that feeds the heart oxygen and blood, or it can be in peripheral arteries, such as the femoral artery. And if there's a plaque buildup, you can get reduced blood flow, or it can rupture and form a complete blood clot as well, which completely stops the blood flow from passing through it. And anything downstream of that is now going to become what's called ischemic. It means it has no blood and therefore oxygen. And if it's not opened back up or replaced with a new synthetic or non-synthetic alternative, it's going to die. So if you have a blocked leg artery, you know that can cause your leg to be amputated. And if you have a blocked heart vessel, then that can cause part of the heart to die, or the whole heart can die and thus be fatal. Other reasons why arteries can be damaged are things such as genetic diseases, such as Marfan's, Ehlers-Danlos, Loeys-Dietz, and aortic aneurysms that form from these different genetic diseases. And these diseases are diseases of the connective tissue, so the kind of main constituents that give your blood vessels its elasticity and its strength and its ability to stretch and go back to where it was. And when you have problems with them, then the actual artery can become damaged in various places, especially where there's high pressures and high blood flow. And then last but not least, you've also got just simply having hypertension. So that means high blood pressure. High blood pressure can cause damage of your arteries, as well as obviously trauma. So if there is some sort of physical trauma to your arteries, here we've got someone who's broken the femur, which could cause femoral artery dissection, which is very, very dangerous. But many different reasons that you could actually have a trauma to one of those blood vessels and need it being replaced. So what I'm trying to say here is there's a whole huge range of different reasons why you might have damage to one or more of your arteries and they need to be replaced. Now, what do we do now? What are the current replacements, and are they good? Are they bad? What are the pros and cons that we've got to them? So I guess the gold standard that you would want to have an artery replaced with is another artery. So you can actually have a donor blood vessel from a different part of your body, and you can use that to replace the damaged artery in the other part of your body. So typically these can be internal mammary arteries, radial arteries in your arms, and also these saphenous veins that are in your legs. And they obviously have a very good advantage in that they have the exact same stiffness and elasticity as the artery that's being replaced. That is said when you're actually using arteries themselves. If you're using saphenous veins, not quite the same elasticity, but getting pretty close. They're not highly available at all. This is a big con that comes with these, in that you only have a limited amount that you can use, and you know, you're going to cause damage to that site and you can't reuse them. So you only have a certain amount of these arteries that can be used to replace damaged arteries, and typically done for coronary artery bypass grafting. So that's heart bypass, as you might know it. They cause damage at the donor site. So obviously where you take this out, it's going to cause trauma and need to undergo healing. And for the most part, like the arteries don't become blocked, but the saphenous veins can become blocked. And they can last for a certain number of years. But if they do become blocked, then obviously they need to be replaced, and you need to undergo surgery, and it can be dangerous if they do become blocked. So yes and no, do they become blocked? And they don't encourage new blood vessel growth, but that's probably not so much of an issue because you've got a new artery sitting there. But they're unlikely to remodel into the kind of format that your native artery already has. So what can we use instead of donor blood vessels? Well, we can use synthetic materials. And typically, we use Dacron for larger vessels, such as the aorta, which is the largest vessel that comes out the heart, and Gore-Tex, or we call it EPTFE, polytetrafluoroethylene, but you might know it as Gore-Tex, which is the same plastic that we make water bottles and raincoats out of. Can be used to make these grafts of a smaller diameter. They are much stiffer than your native artery. They don't have a similar stiffness to your artery. They're not really stiff, like they're not horribly stiff, but they're too stiff in terms of matching to the actual artery. At high blood flows in the aorta, this is not so much of an issue, but at smaller diameters, this is quite a big issue. We can make them easily. Anyone can make them very easily in bulk, and we can make a lot of them. And they obviously cause no damage at the donor site because they're completely synthetic. Now, do they become blocked? Yes, they do become blocked. Again, the larger replacements made out of Dacron for the aorta, they don't become blocked very much. But when you get down to smaller diameters, and specifically 6 millimeters and below, they get blocked a lot. And it's not a very good alternative. And you can imagine if it gets blocked, that's a bad thing. That's a really bad thing. You're going to have you're going to have to come back in and have another replacement put in, another surgery, which is not ideal at all. Or, worst case scenario, you could the blockage could cause you to have an amputation in your leg, for instance, if it was in the leg artery. They are permanent synthetic replacements. So they're just a plastic that's put in your body, and you hope that your body ignores it and doesn't degrade it and just leaves it as it is, and it doesn't replace any of the tissue that was damaged in the first place. So what are we trying to do? We're trying to create a material that answers some or all of these questions and has advantages over especially the synthetic materials that are already out there, but hopefully matching or being more improved over the donor blood vessels that you can get out there. So this is through a collaboration with a postdoc called Ziyu, Dr. Wang, and also Professor Tony Weiss, who I've worked with for a long time, and so has Paul. Paul's worked with Tony for a long time as well. And these are very talented biomaterial scientists and tissue engineers at the University of Sydney. And they created this lovely artery replacement made out of tropoelastin and this polyglycerol sebasate. And tropoelastin, which is one of the materials it's made of, is a highly elastic and degradable protein. So it's naturally made within your body. We can actually make it in bulk in the lab. And this tropoelastin is the monomer to the polymer elastin. So lots of tropoelastins stick together to make elastin. And elastin is the stretchy protein that makes your skin stretchy, your blood vessels stretchy, your lungs stretchy. So it's a very highly elastic protein that we can use. And also the body can break it down, and it's non-toxic because it's naturally occurring. Polyglycerol sebasate, PGS, is also highly elastic, and it also degrades into non-toxic monomers. So basically the polymer, the kind of plastic that we make, when it degrades, your body just absorbs what it's degraded into, and essentially you just excrete it out the body. So it's completely degradable, and it will just disappear if it sits in your body for long enough. So what we did is we actually weaved together nanofibers of these materials to create a highly elastic and degradable graft, essentially. And if you can imagine how small a nanometer is, it's essentially 1,000 times smaller than a millimeter. So they're very, very, very small, tiny little fibers made using something called electrospinning. So this is a picture of one of our grafts. This 4 millimeters from here to here, the diameter. And when we use electron microscopy, which is a very high-end microscopy technique, which we shoot electrons at the surface to get an idea of what it looks like inside, we can see this nice woven together structure that we can see from the tropoelastin and the PGS. So the first thing we wanted to know is can we actually create something that has the same elasticity or similar elasticness to the native artery? And what you can see here is that Gore-Tex that I was talking about before, which is the gold standard that people would use in the clinic for small vessels, synthetic gold standard that is, it's around 900 megapascals. Now, the higher this number is, the stiffer the material, whereas the native artery is all the way down at 1.5. And you can see that the graphs that they made before in this previous study are much more similar to the native artery. So that was very, very exciting that they could make a nice material that's going to be accepted by the body, that's of a much lower, higher elasticity and much lower stiffness. So already we've answered these questions. We've already found something that is similar elasticity to arteries. We can make it in the lab, albeit not as easily as Dacron and Gore-Tex, but if it has other advantages, then that's fine. And it causes no damage at the donor site, obviously, because it's completely synthetic. So the next questions we want to answer are, can we actually, does it cause blood clots? Does it become blocked as much as the kind of typical synthetic materials used? And does it, if it degrades, obviously as it's degrading, it needs to be replaced by something. And what we're hoping is that as it degrades, it gets replaced by a nice, new, healthy artery. So how do we test whether it's become blocked or not, and test whether it still has blood flow coming through it? Well, one of the first things that we do is we can use this CT angiogram. So you've heard of CT imaging, that means computed tomography. And we have this beautiful instrument downstairs at the University of Sydney that is a CT arm, Siemens Pheno, basically it's called, Artemis Pheno. And this really, really high-tech, lovely piece of equipment allows us to scan the arteries, inject in a contrast dye, and then we can see blood going through our vessels and also through our grafts at the same time. And then we can also use ultrasound to again look within the vessels and see what the blood flow looks like through it and see the different shapes of the vessels themselves. So this is the angiogram that I was talking about before, where we've got a little catheter here. You can just about make it out. This curved thing here with a little kind of tail that's a pigtail, actually. It's called a pigtail catheter. And that's injecting in a contrast dye that's going up out the heart and then up through these carotid arteries. And this study is specifically looking at grafts to replace these carotid arteries. And as I said, we've got ultrasound as well. So we can look at the blood flow through the artery and see if it has this nice pulsatile nature, and see if it becomes blocked or not. And when we did this with Tim, who's a vascular surgeon, we found that if you don't put any graft in, obviously the blood vessel stays open. So this is just a control, just to show the blood vessel is nice and happy. There's no unhealthiness anywhere. It's just acting as it should do. But when we put our control in, of the kind of gold standard synthetic substitute, Gore-Tex, what's used actually in the clinic for small diameter vessels, we found that most of them become blocked. So they actually become what's called occluded, and the blood can no longer flow through them. Whereas with our grafts, very excitingly, about 6 times more of our grafts remained unblocked and remained patent. So the blood was still able to go through it. So this is a very, very encouraging new finding. So we've just shown that the vessels, our new vessels, do reduce the amount of blockages you see over the current synthetic substitutes out there. So next we want to see, does it degrade? Does it get replaced with nice new material? Nice new tissue, should I say? So a previous study that was done with ZU and Tony, they showed that if they made these grafts very, very small in a different model, they were able to show that the grafts completely degrade over 8 months. And then they not only degrade, but whilst they're being degraded, they actually get replaced with nice new tissue, as you can kind of see this kind of schematic over here. And what you're looking at here is the normal aorta and the fibers of elastin that I was talking about and the cells in between. And over 8 weeks, what that looked like, and you could still see some of the graft here in this kind of fluorescent green sort of color up here. And then over 8 months, you can see the graft is completely gone, and we've got these lovely elastin fibers running through the tissue and all these cells in between. But what we did find in this small scale study is that the grafts actually underwent a small amount of expansion that then stabilized. So we wanted to kind of take account for that when we did a larger scale study where we did larger vessels at 4 to 6 millimeters. So how we designed this was that we knew the carotid artery was 6 millimeters, which you can see here, where we've measured the carotid artery, and we can see in all our different groups it's around about 6 millimeters. And so to account for the fact that we thought these might expand a little bit over time, we actually made our grafts at 4 millimeters, so they would expand hopefully to the correct size. What you can see here is one of those angiograms again, and we've got the our graft on the left-hand side here, which is actually your right carotid artery. And you can see here the synthetic substitute here, PTFE, otherwise known as Gore-Tex. So what we found over time is that our 4 millimeter grafts actually did expand to more closely match the native diameter. So you can see it starts thinner and then it gets wider and wider. And we're hoping this will stabilize and not get any wider, and we're hoping these other areas actually open up a little bit so they match the artery on either side a little bit better. Whereas the PTFE or Gore-Tex grafts remain at this thin diameter the whole way through, because they're unable to degrade because they're a permanent polymer that's not degraded by the body. And you can see that here. You can see this is the kind of native artery diameter, and this is where the vessel started at, our grafts. And you can see the Gore-Tex ones just stay the same, and our ones over time degrade and form and expand to more closely match this 6 millimeter, although it's not quite perfect. And we've still got some time to go. We've still got some a few time points left on this study. So basically, we know in the smaller ones, we've got this lovely new artery that forms. We don't know yet that this new remodeling, this new reshaping of our arteries has formed a nice new vessel, but we will have that answer within the next 3 months. We'll actually be able to find out these vessels, have they formed nice new vessels, and the new artery tissue that's formed is the grafts that we made, are they completely gone? Have they been replaced by loads of cells? We're going to have all those answers in a few months, which is very exciting. So what is next? So we've got a PhD student who's going to be joining the project, which is very exciting. We've still got some CT and ultrasound that we're left to do. And we're going to be doing those measurements to look at the new artery that forms and the cells that grow in it. And do they match what the cells look like within your native artery? And in order to do that, we're going to do these techniques called histology, immunohistochemistry, and use microscopy to look at them. So that's the kind of nice fluorescent images you saw before. We tag the different cells with different colors and try and find out, does it look like the native tissue? And we've got these other high-end techniques like proteomics to look at the proteins, metabolomics to look at the metabolites. John O'Sullivan, that I mentioned before, is a metabolomics expert, so that's something we do routinely in the lab. RNA sequencing, which is looking at the mRNA. You might have heard of mRNA before. It's simply the messenger that is the code that allows your cells to create proteins. And we can look at all the different RNAs and see which ones are being upregulated and which ones are downregulated. So does our new tissue have cells within it that have similar mRNA profiles to that of the native artery? And then, as Catherine explained, we're certainly going to be doing this spatial transcriptomics, which is essentially a way that we can get each individual cell and look at that mRNA expression. So does this cell express a lot of this mRNA? Does it downregulate this mRNA? What proteins are the cells trying to make? And we can see all that actually spatially on a microscope slide. So it's a very new, exciting technology, and it gives you the spatial information as well as the mRNA information. So does this cell upregulate this, and does it affect the cell next to it, and et cetera, et cetera. So what's next for the project? Well, we want to try different shapes, sizes, and lengths of these different grafts and see what we can do with that. We want to look at, replacing leg arteries, femoral arteries, and coronary arteries, and eventually scale it up to a much, much larger size and see what we can do replacing parts of the aorta as well. And it's a long way off now, but we're certainly working towards clinical trials, and that's definitely our aim. Is we don't want to just do science for science sake. We want to do translational research, and we want to get things into the clinic to help people with these different diseases and issues with their arteries. So that's it. I want to acknowledge everyone. I obviously want to acknowledge Paul and Sean and John and Lakshay, and of course Catherine for organising everything here. People at the RPA, so we've got a whole host of different clinicians over there that we work with. Tim specifically works a lot on this project and has helped out massively and is a big part of it. Everyone within Tony's lab, so we've got ZU and Tony and Sue, who also helps out making the tropoelastin, and a whole host of people at the university, specifically Hugh also helped out massively with these surgeries, as well as other people in the hybrid theatres such as Lisa and Inez. And last but certainly not least is all of you. I have to thank you for all your kind donations and kind help. Without your donations, none of these experiments would be even remotely possible. So highly appreciate your help, and thank you very much. And please let us know if you've got any questions.
Thanks, Rob. Catherine, you seem to be on silent there, I think.
That's okay. Okay, here I am.
Okay, that was fantastic, Rob.
Yes,
thank you.
Very, very interesting. So you can either there's a little hand at the top where you could hold your hand up if you had a question, and Daria here could unmute you to talk, or you could send there's a Q&A little icon at the top. You can send a question through on the Q&A, and Paul and Rob would be only too happy to answer them for you.
I might just add a line or two after that, if you don't mind.
No.
The reason, some of the reasons behind trying to find or trying to construct a blood vessel, in the laboratory, if you like, is based around understanding how blood vessels work, whether they're small or large vessels, whether they're arteries in your arm or in the feeding blood to the heart, the coronary arteries, or the large vessels of the aorta. We know that blood vessels don't allow stop the blood from clotting within them, but we also understand, but not fully, why that is. There's an inner lining of the blood vessels which makes sure the blood stays thin, if you like. We also think that it's a combination of that. It's an interplay of that lining as well as what we refer to as the biomechanics, which Rob touched on. So how that vessel wall moves with each heartbeat, with changes and fluctuations of blood pressure to maintain what we're after, laminar flow or smooth flow in the vessels. And that affects the distribution of the blood flow to all the different organs of the body. As I said, it prevents blood from clotting. So, but it's not just arteries to make sure those blood vessels keep flowing blood to the different parts. It can also be how the valves of the heart also work. And if we look at some of you I know have had aortic valves or aortic replacements. If we look at the aortic valve, sometimes we implant that your own valve inside a Dacron tube, which is another form of synthetic tube. We know that that Dacron tube is stiffer than the native aorta. And we're also pretty sure that that interplay between the valve that you're born with and with a normally functioning or normally biomechanically elastic and appropriately responsive aortic wall probably allows the valve to last longer. And so the current versions of what we put in there are either a new valve from a pig or a cow on a fairly stiff frame, or we reimplant them into synthetic Dacron tubes. So it may not only be, this work may not only play out to keep blood vessels open longer or replace blocked arteries, but it could also allow us to reimplant heart valves or fashion synthetic heart valves in a more appropriate tube, if you like, and hopefully improve durability. So there's a lot of different possible results from this research. I think understanding biomechanics and maintaining flow through the body. The body does it a lot better than we can currently do, and we're trying to mimic that, I guess, trying to learn about it.
Very good. Any questions there? Anyone want to raise their hand and then we can unmute you just with that little icon at the top, or just quickly type in a quick question? Nothing there. Rob, did you have anything further to say? That's pretty much, you've done a great presentation. It's really been fascinating to hear the work going on there, but anything to end really, and while we wait and see if anyone asks a question?
Yeah, I think there's so much that can be done with these vessels, and there's such a wide range of issues that can be treated with it. And I think the result, just the first result showing that they stay open, they don't get blocked as much as the PTFE, the Gore-Tex. That's just really, to me, it's very, very, very exciting. We haven't finished the study yet. We've got a long way to go to finish the study, in fact, but the end of the kind of main first bit, the 8 months study with the ultrasound and the CT, that's almost finished. But just that first result was very encouraging, and I'm very excited to see what happens and what it looks like under a microscope, and see, have we formed this nice new vessel, and have we just completely replaced it with a new healthy artery? And you can imagine the advantages of just sticking something in and it just grows a new aorta, and then it disappears. So that's the kind of potential with it. The other thing as well is there's other people working on similar things to this, but they often have cells implanted into the graft itself. So they've already put cells into it, and then they put it in, and then it degrades and tries to form a new vessel. Now they're okay. They've found some good results that inferior to the preliminary results we have. And on top of that, you can imagine, as a treatment, that's a very, very difficult thing to get to someone. It's very difficult to get that graft, if you've made it in a lab and you've put some cells in it, to have it very, very regulated and very, very similar each time. It's going to be quite difficult. And getting it all the way out to different communities, especially rural communities, it's going to be very hard. Whereas our ones, you literally can put them in a shelf. They don't even really need to be refrigerated. They should be refrigerated, but you don't have to be. So As long as you can ship them on some ice, you can get them to wherever you want, and they can just be made the same each time. So they hold a lot of advantages over even the kind of things that people are testing at the moment, let alone what's actually being used.
Amazing. Okay, well, I think there's no questions here. I can't see any. But we'll keep you updated on our research. We'll tend to do these webinars 2 or 3 times a year, but always we'll just keep you updated on what's happening. Things don't move really fast in the world of research, but we will definitely have another webinar down the track on this topic for Rob to update you on where things are at, just so you know what's happening with your money and when you donate, how it's helping many people. In the future, it's going to be able to help lots of people with this particular research, I can see. So I can't see any questions, so I think we might finish up there. We will send a recording. I'll send out a recording in the next few days to everyone who's registered, whether you're here today or not. So if you were here and you wanted to go back over it, you can watch it at your leisure and pause it when you wanted to find out what Rob was saying in particular parts or looking at some of his slides. So thanks all for joining today. Really appreciate you your interest and your support, and we'll look forward to seeing you again down the track. Bye-bye, everybody.
Thank you.
Bye-bye.
Thanks. Thank you, everyone.